YouTube. Today we're focusing on meiosis, the cell division that creates life's diversity. This is based to paper 2 also. So before we do, don't forget to like, share, subscribe, and follow for more based to in life science. What is meiosis? Meiosis is a specialized type of cell division that produces gametes, sperm cells in male and egg cells in females. Unlike ordinary cell division, meiosis reduces or minimizes the chromosome number by half, transforming diploid cells into haploid cells. Diploid cells means 2. Haploid cells means 1. So this process is absolutely essential or important for sexual reproduction and serves as the engine of genetic diversity. Without meiosis, offspring will double the chromosomes of their parents with each generation. It creates reproductive cells, gametes, halves, chromosomes count 46 to 23 in humans, generates genetic variation in offspring, occurs only in reproductive organs. The meiosis is a cell division whereby all diploid cells, as I said, somatic cells, undergoes to two cell division to form 4 haploid cells or sex cells. Somatic cells, all body cells except sex cells. Diploid, 2 set of cells, chromosomes. Haploid, 1 set of cells, chromosomes, as I mentioned earlier. So I have the structure with me here. I will post it on and at the end of this chapter, so then it can be written. But let's continue. Overviewing 2 divisions for cells. There is meiosis 1 and meiosis 2. Meiosis is unique because it involves 2 consecutive divisions that produce 4 distinct daughter cells from a single parent cell. This double division is what makes meiosis fundamentally different from mitosis. Here it goes. Meiosis 1. Homologous is the pair of chromosomes separate, creating 2 haploid cells, double haploid cells. Each cell still contains duplicated or double, chromosomes, sister chromatids joined together. At first, meiosis, we have prophase, metaphase, anaphase, telophase, and then it is a prophase 1, metaphase 1, anaphase 1, telophase 1. Then there is meiosis 2. Sister chromatids separate, producing 4 genetically unique haploid cells. These become the functional gametes ready for fertilization as the gamete as sex cells. And then on prophase 1, I don't have the structure of prophase 1 on the screen. Prophase 1, I will give the hints of the prophase based to it, but the spindle fibers and the nucleus, nuclear membrane, you can utilize your study guide, but they have these points which can make it differ from each of the phases. At anaphase 1 or on anaphase 1, spindle fibers shorten and pull the homologous chromosomes to the opposite poles or direction. Then on to anaphase, there is central spindle fibers chromosomes, homologous chromosomes. The next, the first one it was anaphase, but the first one is supposed to be prophase. The prophase, there's a spindle fiber, a nuclear membrane. This is the first phase. The prophase, anaphase, is the one with the prophase is the one with spindle fibers, nuclear membrane. At the prophase one, it is the formation of the is the beginning of those phases before the other ones or the following three phases. Anaphase 1, it has central spindle fibers, homologous chromosomes. And then anaphase 1, it has spindle fibers shorten and pull the homologous chromosomes to the opposite poles in direction. It means there is only 4 chromosomes but separated from each other but being pulled with the spindle fibers. And then telophase, my bad. Cytokinesis takes place here. A cell will divide into two. It also explains itself that there is two cells splitted with 2 chromosomes together at each cell. Cytokinesis is the division of the cytoplasm. And then we have metaphase. Is this a plant? Oh my god! These are the questions that were given, or to differ these things. Metaphase is this is a question. Is this a plant or animal cell? The answer it was animal cell. There is a cell membrane without a cell wall. That's the differ between the plant and the animal cell. Then there is a centriole where spindle fibers are attached, and the plant spindle fibers are attached in the cytoplasm. This makes the difference between the animal cell and the plant cell. Is this a human cell? Then this is another question. Is this a human cell? Give a reason for your give a reason for your answer. No, it did not. And the reason I almost forgotten to return this thing, but at prophase 2, the pair of chromosomes are split into 2. Metaphase 2, the homologous chromosomes are aligned on the equator at both cells. Anaphase 2, the centromere break and the chromatids more to the opposite poles. The spindles shorten and pull at the cells. The spindle fibers are pulled to the opposite side of the cell. Telophase 2, the chromatids are in 4 cells, which are called daughter cells. The difference between meiosis 1 and meiosis 2. Meiosis 1, crossover takes place. Meiosis 2, no crossover. Meiosis 1, homologous chromosomes are aligned in the equator. And the meiosis 2, individual chromosomes are aligned in the equator. At the end of two cells are formed in meiosis 1. In meiosis 2, 4 cells are formed. In meiosis 1, chromosomes are 4 at the end. Chromatids remain the same. Diploid and haploid chromosomes. Then we have the human sperm. Human sperm has 23 haploid chromosomes. An ovum or ova has 23 fertilized ovum, a diploid zygote with 46 chromosomes, which means the ova has 23 haploid chromosomes. When it goes to process of fertilization, the ovum has a diploid zygote which forms the zygote with 46 chromosomes. As we all know that the chromosome number 1 to 22 are called autosomes. Chromosome number 23 is called genome. Genome determines the sex or gender. Female chromosomes are XX with the same side, same shape, and the male chromosomes are XY, which determines the male gender. X is bigger than, but at the male, the X is bigger than Y. The karyotype above represent, as I was explained to you, that the X is bigger than chromosome Y, or at the table can be illustrated as one tip and the small tip, which determines the male gender. When the tips or the shapes of the chromosomes are in same size, it means it is a female gender. Genetic variation. Variation means a different shape, number of all of alloses and shape within an organism. A variation is a difference. Yeah, this information is based to what I've revealed to you, in which it says meiosis 1, prophase 1, chromosome condensation, but the ones I've mentioned, they are much easier and simple and understandable. So here you get, then it says metaphase 1 to metaphase 4 to metaphase 1. Metaphase 1, homologous chromosomes pairs tentrate or align at the cell's equator, as I mentioned it earlier. Yeah, I covered everything before getting to this topic. There is the information with me here. After a brief interface with no DNA replication, the two blood cells enter meiosis 2. The second division represents mitosis, but occurs in cells that are already haploid. Haploid, it means one set of chromosomes. At prophase 2, chromosomes condense once again in each of the two haploid cells. Spindle fibers reform and attach to the centromere of sister chromosome. The centromere is the is the cycle like at the between the chromatids, which is when there are two or from when the two chromatids form the one chromosome, which is at the center of the chromosome. It is called centromere. Metaphase 2. Individual chromosomes, still consisting of sister chromatids, line up single file at the cell equator, similar to mitosis. Metaphase. This much is written here. It is clearly. I already mentioned this information, but let me highlight you. The final stage of meiosis complete the gene from one diploid cell to four haploid gametes. This is where sister chromatids, which have been together since DNA replication, finally separate. The anaphase 2, centromere split and sister chromatids separate, pulled to opposite poles as individual chromosomes. Cytokinesis, cell division completes, reducing four genetically unique haploid daughter cells gametes. Telophase 2, nuclear membrane reform around four haploid nuclei. Chromosomes begin to decondense to deform. Each of the form resulting cells contains half the chromosome number of the original parent cell and is genetically distinct from all others due to crossover and independent assortment. Why meiosis matters? Genetic diversity. Meiosis is the biological foundation of genetic diversity. The variety or more of genetics information within a population. Two key mechanisms create this diversity as crossover and independent assortment and the genetic diversity. During prophase 1, homologous chromosome exchange DNA segments, creating new combination of alleles on each chromosome. And also know that alleles contains DNA portion, DNA with portion within the portion, independent assortment. During the metaphase 1, chromosomes pair orient randomly, creating 23 here to be diploid chromosomes. 23. 8.4 billion position combination into women, which is called the zygote. These two represent diploid chromosome. It means it's 46 chromosomes, which forms the zygote. This variation explains why siblings look different. Here is explained at the first chapter of DNA, the powers, natural selection, and the evolution, and enable species to adapt to changing environments. Without meiosis, there would be no biodiversity. Key points to remember. Those are the key points to remember based to this meiosis topic. Now we are wrapping up the topic. So this much essential to know these things, and I gave some points. Let me give you many points. So everything is there for paper 2. Also watch the video I've posted, which is based to DNA, the code of life. Thank you for watching this video. You found it essential for your grade 12 learners or candidate. It will help you to succeed your marks on your paper for paper 1. I will also drop many more videos for math, life science, agriculture, and math literacy. So make sure you stay on this channel. Don't forget to like, share, subscribe, hit the notification bell, and so every time I drop, you get notified. Don't forget to like,